So far in this biology class, we've been looking at how life is built from the bottom up. And we've looked at how the building blocks themselves are built from atoms. And then the building blocks make up other things. For example, the proteins we've looked at already. We've looked at how DNA and RNA is built. And today we're going to look at how the lipid membranes are put together. Before we do that, let's talk a little bit about boundaries, since the lipid membranes form the boundaries for the cell. Boundaries is something that I consider an essential to life, but not everybody considers it like that. Most people agree that you need information, you need some sort of machinery, and you need energy, but what about boundaries? Couldn't there be another way to keep everything together? Well, the cell uses little tethers to hold on to things that need to be held on to and kept in one area. But the unfortunate thing about tethers is that if everything was tethered, it might all get tangled up. So that might not work so well. Also, the cell uses DNA or other molecular fibers as kind of a one-dimensional boundary. When something binds to DNA or binds to another fiber in the cell, it's held in that one-dimensional line until it lets go. So that would be one way to keep things together, but the problem there is that things ultimately do have to hop off and hop back on. And if that hopping off wasn't kept in some sort of a boundary or enclosure, it would just float away and never come back. So that wouldn't work for everything. You could imagine a boundary being a two-dimensional surface. That might be okay, and if everything just stayed on the surface, then everything would be always in one place. But it limits things, just like if you've ever read a book called Flatland, shapes that are stuck in flatland can't experience the full joy of being in three dimensions. So that would be not so good for the cell. So basically all of life uses three-dimensional enclosures, and these enclosures always include a lipid component to them. So that's why we're going to learn about that today. Now, in addition to sort of just keeping everything together, there are other reasons for having these enclosures. One big one, which we'll be talking about today, is that when you put a boundary or an enclosure around something, you can have a gradient that creates a driving force. And we're going to learn about that gradient. It's kind of just like having a battery with a plus end and a minus end. Another reason to have these is to keep larger cells broken into bite-sized chunks. We've already hinted at that, and we're going to look at that later on in this course. Another reason is just that enclosing one group of reactions, chemical reactions, in one place is important because those reactions might be incompatible with other reactions. For example, the cell has to digest things inside of it, so it has these little enclosures that are at low pH, but low pH is not compatible with the rest of the cell, so it's important to keep that separate. So let's talk a little bit about the driving force. I mentioned that this is like a battery. So what is it that makes a battery go? It has a plus end and a minus end, and that's because there are little electric charges. We call them ions. And the ions are at one end or at the other end. If they're a plus ion, they might be at the plus end. If they're a minus ion, they might be at the minus end. You can experience the power of those ions when you attach the battery to a circuit and look at it light up a flashlight, or some people might even just take the battery, if it's a 9-volt battery with the two ends right here, and lick it and experience the power of the battery right on their tongue. I don't recommend that, but you can definitely do that. The car batteries that we have in our cars are even huger, and they have their two ends sticking up. And it's very important that you don't connect a jumper cable right from one end straight to the other, because you will get huge sparks, huge releases of energy. And what this is doing is it's wasting the ions and the power of the ion flow. So the cell does not want to do that. The lipid boundary is there to prevent all those ions from quickly going from one side to the other in the cell. And the lipid bilayer is very nice because it prevents positive or negative charges from going across. The lipids, as you can imagine, are kind of greasy. That's why foods that are greasy are high in lipid content. And if you're trying to watch your lipids, you don't want to eat those foods. So the greasy layer prevents things that are more watery or charged from going across it. Just like in salad oil, like we were talking about earlier. Water and oil don't like to mix. And water loves things like ions. That's why you can have ionic fluids inside of your water. You might get charged up with your electrolytes, drink some Gatorade. Those things all dissolve really nicely in water, but they do not dissolve into oil. And so the lipid membrane creates a nice boundary around the cell. In fact, if you would like to kill a cell, you can put a hole in the membrane, and that will just kill the cell because it will allow all those ions to just flow across, and there won't be any more power to power up the cell. So that's one way that the immune system kills enemy cells. Now, there is a particular kind of motor in the cell that looks like an outboard motor. It's a rotary motor, and it is powered by the same ionic flow. You might not think that inside of cells there could be a rotary motor. You look at the animal world and you don't see anything that has a wheel on its side. There's some snakes and there's some worms that curl themselves up into a ball and roll, but there's nothing that actually has like an axle going into its torso and rolling around like a car. But at the cellular level, there are these outboard motors. There are axles that spin freely inside, and there are all the components of an electric motor. One of these is called the bacteria flagellum, and it powers bacteria through the fluid. And the way it gets its power is by allowing little ions to flow through what looks like a turbine. So a turbine moves around and around as water or steam pushes through it. And instead of water or steam forcing its way through these little molecular turbines, there is little ions that turn the turbine around and around. So does it go clockwise or does it go counterclockwise? Well, the interesting thing about this bacterial flagellum motor is it can do both. And because of the natural twist of the little corkscrew-like shape that's on the outside of this, if it twists clockwise when viewed from the top, then the bundle of fibers that makes up the corkscrew flies apart, looks like somebody's touching a Van de Graaff machine and their hair is standing up on end with static electricity. They just go all over the place, and they're not useful in screwing the bacterial body through the cell, through the fluid that it's in. So that doesn't really work very well in terms of powering it, but when it spins counterclockwise, all those twisty fibers bundle up into a nice, solid corkscrew-like shape, and it just screws itself through, propels itself through the fluid perfectly. So that little switch going between clockwise and counterclockwise is essential for bacteria as they find their food, and we'll discuss that in the next lecture. So these are interesting features of having a membrane and keeping a gradient of ionic charges on one side but not the other, so that when a little hole, a controlled hole is released in something like a turbine, they can flow quickly across and power the turbine in a circular motion. Now, if you're doing a little bit of textbook reading from BioBook, the assignment for today is to read about what are lipids, which is a review from last time. And you can read also about how cells maintain separate internal and external environments, and also how ATP synthase makes ATP, because that's another rotary motor. So pay attention to how the proton gradient in ATP synthase works to power ATP synthase.